The central dogma explained how living cells process information. When we talk about how cells work, it's important to see them not merely as a gelatinous bag of chemicals, but as fully functioning chemical computers. In many ways, living cells are nature's analog to man's computational devices. All cells, except for red blood cells, fulfill the general architecture of a computer. A computer receives an input from the outside world and processes that data with programs stored in the hard drive. That program then acts on the data to produce a meaningful output. Likewise, living cells receive signals from the outside world, process that information through protein mediators, and use the genes stored in DNA to produce an appropriate response. How a cell receives and transmits these signals is a story for another video. Instead, our purpose is focused on the second half of this process. The way a cell uses genes to create a functioning protein that then outputs a response is known as the central dogma of molecular biology. Part 1. DNA stores the machine code for living cells. Most DNA can be found in a cell's nucleus. The rest can be found in the mitochondria, the powerhouse of the cell. When the cell is dividing, you can see DNA as long X-shaped sausages, called chromosomes. During normal functions, DNA is spiraled out as free-floating strands called chromatin. DNA is the medium living cells use to store its genome. Just as a hard drive stores files and programs for computers, DNA stores the genetic code that tell your cells how to make you. The key difference is that DNA uses a quaternary system to store genetic information. This means that the 4 nitrogenous bases, adenine, thymine, guanine, and cytosine can each code for 2 bits of information. For example, A equals 0, 1, T equals 1, 0, G equals 0, 0, and C equals 1, 1. So how much data does your cell store? There are 3.088 billion base pairs and 23 unique chromosomes, plus the DNA in your mitochondria, since a human cell typically carries 2 of each chromosome, except for the sex chromosomes. The total storage space of a cell is 8.84 billion base pairs, or 17.68 billion individual bases. Each base is worth 2 bits, and there are 8 bits in 1 byte. Final calculations show that every human cell has 4.42 gigabyte of storage space. The human genome, which is all the genes found in human cells and the non-coding regulatory regions, consists of around 6,469.66 megabase pairs, which translates to 12,939.32 million bases. Once again, each base is worth 2 bits, and there are 8 bits in 1 byte. Multiplying all that together gives us a grand total of 3,234.83 million bytes, or 3.23 gigabytes. This means that your genome makes up about 73% of each cell's memory. The rest are DNA segments with no known functions. Usually, they are junk DNA or fragments of ancient genes long since degenerated. Another important difference between hard drives and DNA is the absence of a writing head. As of the creation of this video, there is no known naturally occurring system of writing a new gene from scratch. Instead, genetic information is acquired through elaborate copy and paste mechanisms and other chemical processes that cannot possibly be covered in a single short video. By themselves, DNA are just useless strings of chemicals, and hard drives are useless disks of silicon. Just as a computer needs a CPU and RAM to retrieve and store data, a cell needs a system that can copy the genes stored in the nucleus and create a short-term working memory. Next time, we will take a look at the 2-step process that turn genetic code into flesh and blood: transcription and translation.